Good morning, children. Till our last class, we have seen about Mendelian genetics, that is about monohybrid, dihybrid crosses, and you know very well what is meant by monohybrid and what is meant by dihybrid. And he only gave an initiation for the factors called genes. After his discovery only, and people and other scientists came to know that characters are controlled by genes, and he explained with some pea plant experiment. According to his discovery, at each phenotype of an individual are controlled by a single gene, and each gene consists of two alternative alleles. Say for example, when we have a height of plant as tall and dwarf, the tall plant consists of alleles like capital T, and dwarf plant consists of alleles small t. So this was about Mendelian genetics, okay. But now we are going to see is extensions of Mendelian genetics. What does mean by extensions of Mendelian genetics? It is nothing but after Mental period, the things have discovered based on Mendel's discoveries are called extensions of discoveries, extensions of Mendelian genetics. In this, according to these concepts, the phenotype of a character is controlled by more than two genes, and each gene consists of two or more alleles. When these two genes interact and control the phenotype of character, and this is called what? Genic interaction. This genic interaction are of two types. One is intralocus genic interaction and interlocus genic interaction. What is mean by intralocus genic interaction? See, what is mean by locus? Locus is nothing but the place where the particular genes are located in the chromosomes are called loci. Suppose if the gene interaction has taken place in the among the genes which are present in the same locus are called intralocus interaction. Interlocus interaction, if the genes are present in the separate locus of chromosome and interaction has taken place between them, and it is said to be interlocus interaction. Okay, in this there are many subdivisions are there, that all we will see by our text. Okay, extensions of Mendelian genetics. Apart from monohybrid, dihybrid and trihybrid crosses, there are exceptions to Mendelian principles. That is the occurrence of different phenotypic ratios, the more complex patterns of inheritance, or the extensions of Mendelian genetics. There are examples where phenotype of the organism is the result of the interaction among genes. Gene interaction. A single phenotype is controlled by more than one set of genes, each of which has two or more alleles. This phenomenon is called as gene interaction. Okay, a single phenotype is controlled by more than one set of genes, and each set of gene has two or more alleles, and this phenomenon is called as gene interaction. Many characteristics of the organisms, including structural and chemical, which constitute the phenotype, or result of interaction between two or more genes. Gene interactions. One is intralocus interaction. It is called allelic interaction. Then interlocus interaction, and this is called non-allelic interaction. That is allelic and non-allelic interaction. Actually, alleles are located in the same locus of chromosome, but non-alleles are located at different chromosome, different loci of the chromosome. That's what it is called allelic interaction and non-allelic interaction. And then you can see this interlocus interaction are of two types. One is epistatic, another one is additive. Epistatic and non-epistatic interaction, additive and polygenic interaction. And about intralocus interaction, there are three main types. One is dominant relationship, lethal genes and multiple alleles. In dominant relationship, there are four types. Complete dominance. For this example, what we have discussed in Mendelian genetics, that is monohybrid, cross, tall and dwarf, is an example of complete dominance. How could we say like that? When mental crossed the pure breeding tall and pure breeding dwarf, what happened? In the F1 generation, all the plants were tall only. All the plants were tall only. So here only tall characters are expressed and dwarf character did not expressed in the F1 generation. Hence here the tall is completely dominant over the dwarf and it is said to be complete dominance. And incomplete dominance we are going to see an example in 4 o'clock plant, Mirabilis chelaba. And in then codominance. What is codominance? It is our blood group is an example for codominance. See, we are having different types of blood grouping among the human. A blood group, B blood group, A, B and O blood group, isn't it? If a person has allele for A or AO and he has a blood group A type. Suppose if he has alleles of B, IB or IBIO and he has the blood group of B. But when he has alleles of IA and IB, one allele consists of allele IA, another one is IB. When the two alleles are present together and the among these two alleles, none of the alleles will dominate each other. Okay, so both are together expressed in its organisms. Okay, so AB blood group will the person will get. Okay, and this is called what? Codominance. When the two alleles, when two different alleles are expressed together in an individual, it is called codominance. And over dominance also, that is it is another example for that. And lethal genes, lethal means what? It causes death, that is called what? Lethal genes. In lethal genes, dominant lethals, recessive lethal, conditional lethals and sex linked lethals. If dominant gene is responsible for the death, it is called dominant lethals, and the same way recessive and sex linked also, and balanced lethals also, totally 5 types and multiple alleles. Let's enter into the text. Mendelian experiments proves that a single gene control one characters, but in the post-Mendelian findings, various exceptions have been noticed. As I said, a Mendelian experiment proved that a single gene controls one character, but in the post-Mendelian findings, various exceptions have been noticed in which different types of interaction are possible between the genes. This gene interaction concept was introduced and explained by W. Watson. This concept, otherwise known as factor hypothesis or Batson's factor hypothesis. According to Batson's factor hypothesis, the gene interaction can be classified as intragenic gene interaction or intraallelic or allelic interaction. And another one is intergene interaction or interallelic or non-allelic interaction. Okay, the intragenic interaction hypothesis was first discovered by and explained by W. Batson. Hence, it is also called as Batson's factor hypothesis, or otherwise known as factor hypothesis. And there are two main types in that. Let's enter into the intragenic interaction. Interaction takes place between the alleles of the same gene, that is alleles at the same locus, is called intragenic or intralocus gene interaction. Already I said, if genes are having a particular loci in the chromosomes, okay, genes are having particular locus in each chromosome. So each gene has two or more alleles, right? So when the genes are present in the particular loci and its alleles also present in that same locus itself, whether it is two alleles or three alleles, when the interaction has taken place among these two alleles of the same locus, it is called what? Intragenic interaction, okay? And it is divided into four types: incomplete dominance, codominance, multiple alleles, pleiotrophic genes are common examples for intragenic interaction. Then incomplete dominance, no blending of genes. The German botanist Karl Koren's 1905 experiment in 4 o'clock plants Mirabilis jalapa, when the pure breeding homozygous red, or its phenotype, its allele is capital R1, capital R1, parent is crossed with homozygous white, its phenotypic is capital R2, capital R2. The phenotype of the F1 hybrid is heterozygous, that is pink. It consists of capital R1, capital R2 as its allelic form. When in this, what we have to observe when compared to Mendel's monohybrid cross, see, please recall Mendel's monohybrid cross. In Mendel's monohybrid cross, when he crossed pure breeding tall and dwarf, and they all F1 as tall plant, that is, it express one of the parental trait. But in this incomplete dominance, the German botanist Karl Korenz, he explained the experiment in 4 o'clock plant Mirabilis jalapa. He crossed two pure breed, one is red, another one is white. When he crossed these two pure breeding, in the F1 generation, all the plants are pink. It did not express any of the parental trait, that is either pink or white, okay. So the F1 heterozygous phenotype differs from both the parental homozygous phenotype. This cross did not exhibit the character of the dominant parent but an intermediate color pink. As I said, in the F1 it is heterozygous, it is not a homozygous, it is not a capital R1, capital R1. It has a capital in F1, capital R1 and capital R2, or the two allelic forms in it. So this is called what? Heterozygous. This heterozygous phenotype did not express any of the parental trait. It is different from their parental homozygous phenotype. The cross did not express the character of dominant parent, but an intermediate color pink was expressed. When one allele is not completely dominant to another allele, it shows incomplete dominance. Such allelic interaction is known as incomplete dominance. Okay, so here neither of the allele is dominant over the other allele, isn't it? That means capital R1 does not show any dominance over the capital R2. Same way, capital R2 does not show any dominance over the capital R1. So here both the alleles does not show any dominance, and it is called incomplete dominance. It is called incomplete dominance. ரெண்டு அல்லிலுமே அவங்களோட டாமினன்ஸை யாருமே எக்ஸ்பிரஸ் பண்ணல. ஸோ அதனால இதை நம்ம என்னன்னு சொல்லுவோம்? இன்கம்ப்ளீட் டாமினன்ஸ் சொல்லுவோம். அண்ட் திஸ் allelic interaction is known as incomplete dominance. F1 generation produces intermediate phenotype pink colored flower. When pink colored plants of F1 generation were interbred in F2, both phenotypic genotypic ratios were found to be identical as 1:2:1, one red, 2 pink, 1 white. So in this cases, in this experiment, when we have a look of monohybrid cross of mental, its phenotypic and genotypic ratio is different. Phenotypic ratio is 3:1 and genotypic ratio is to 1:2:1. But in this incomplete dominance of Mirabilis jalapa plant, both phenotypic and genotypic ratio are same, that is 1:2:1. That is one red as of phenotype, one red, 2 pink, 1 white. As of genotypic, capital R1 is 1 and capital capital R2 is 2 and capital R2 is 1. So the genotypic ratio is 1:2:1. From this we conclude that the alleles themselves remain discrete and unaltered, proving the Mendel's law of segregation. The phenotype and genotypic ratio are the same. So there is no blending of genes. Okay, as Mendel said, law of segregation. See here children, from this we conclude that alleles themselves remain discrete and unaltered. Alleles will not be altered. They are being as such without any alteration, and proving the Mendel's law of segregation. The phenotypic and genotypic ratio are the same. There is no blending of genes. If there is a blending of gene, we could see some other combinations. In the F2 generation, R1 and R2 genes segregate and recombine to produce red, pink and white in the ratio of 1:2:1. R1 alleles actually code for an enzyme responsible for the formation of red pigment. So, the allele R1, which is responsible for the formation of a pigment red color, and R2 allele codes for defective enzyme. So, and this one R2 is an allele which code for defective enzyme. R1 and R2 genotypes produce only enough red pigment to make the flower pink. So, the two R1 are needed for producing red flowers and two R2 genes needed for white flowers. If blending had taken place, the original pure traits would not have appeared and all the F2 plants would have pink flowers. It is very clear that Mendel's particular inheritance takes place in this cross, which is confirmed by the reappearance of original phenotypic in the F2, right? Okay, here we are saying that there is no blending of genes. How could we say if all the genes or both the genes R1, R2, both the genes are blending, what will be happen? All the F2 plants will be in pink color. Then there would not be any expression of pure breeding plants such as red and white. But we could see the parental traits of red, white and also pink also. So it is clearly proof that there is no blending of genes in between the R1 and R2. Actually, the the red color of the flower is due to the pigment red, which is controlled by an allele called R1. The two alleles are necessary for the red color of the flower. The two allele R2 is inhibiting enzymes, so it does not express any color. So white in color this flower. Okay, but in F1 generation, one R1 and another one is R2. What happened? Only half white and half red. Then the resulting flower is what? Pink in color. That's what is saying here. And R1, see the diagram, the flow chart children, red, and R2 white. It is a parental generation. And when the when these two pure breeding parental flowers are crossed, the F1 will be appeared as R1, R2, which are pink in color at its intermediate phenotype. When R1, R2 are selfed, what will be the result? 1 red flower, 2 pink flower and 1 white flower. And the ratio is in the F1 generation is 1 R1 R1, 2 R1 R2 and 1 R2 R2. This is what the result of incomplete dominance. This is what is mean by incomplete dominance. It is nothing but any one of the parental trait will not be dominant over the other allele of the character, and this is called what? Dominant incomplete dominance. It can be expressed in the Mirabilis jalapa plant. Okay, next one is codominance. This codominance I will discuss in my next class, children. Today I have taught only one topic, that is interaction, genic interaction. What is genic interaction? And tablates its various type. This is one question. And explain the incomplete dominance with an example, or explain the type of interaction has taken place in the 4 o'clock plant or Mirabilis jalapa. This is the only one, these are the 2 questions you will get as a homework, children, and I will get you in my next class. Bye.